This is TWIS. This Week in Science, episode number 816, recorded on Wednesday, March 17th, 2021. How do Irish eyes? Hey everyone, I'm Dr. Kiki, and tonight on the show, we will fill your heads with bad air, destruction, and a cosmic shipwreck. But first, thanks to our amazing Patreon sponsors for their generous support of TWIS. You can become a part of the Patreon community at patreon.com/thisweekinscience.
Disclaimer, disclaimer, disclaimer. Those who control the present control the future. We current humans are here but for a short while. When we are gone, the world will move on with new humans. New humans with new ideas, new solutions to old challenges, and new challenges of their own. But we are here now, and the future, even the future we will not be here for, is very much ours to see. While we are here, it is a future we can do something about. We may not feel the responsibility of the moment. We may not be so altruistically inclined to sacrifice work and invest in causes beyond our immediate need. But if we do not act, if we do nothing, we will have no say in how our history will be written. Those who control the present control the future, and nowhere are the signs of what is at stake greater than here on This Week in Science. Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening, what's happening, what's happening this week in science. What's happening, what's happening, what's happening this week in science.
Good science to you, Kiki and Blair.
And a good science to you too, Justin, Blair, and everyone out there. Welcome to another episode of This Week in Science. We are back yet again to talk about science. Hey, happy St. Patrick's Day, everybody.
Oh, yeah, that's why I'm not wearing any green.
Something about snakes and
again.
Ireland. Yay!
Yay. I'm going to keep
I'm sure they were perfectly nice snakes. They were just minding their own business, but, you know.
Well, you know, they were minding their own business until the Irish were like, get out of here, you snakes.
Yeah.
We don't need any of those. But I'm sure their ecosystem still would appreciate a few snakes. However,
They didn't get them all. There's no way.
no, they didn't get them all.
Wait,
What?
Did they ever
What?
have snakes to begin with?
Yes,
I thought
of
it was
course.
'cause it was- I thought it was 'cause it
Snakes
was-
everywhere.
I thought it was because it was like some island that didn't have snakes, and then there was a story. It's like, oh yeah, you know how that came about. Why? I don't know. There's this guy who like chased them all away. That's not it. They have snakes. Now we have to figure. Top of the show. First question: does Ireland even have snakes?
Yes, of course they have snakes.
I don't know.
It
this.
has lots of snakes. Oh, wait.
Huh?
Well, there are snakes. They've probably been brought there by other species.
Oh, they're invasive. Okay. So there was. So the myth has its origin in the fact that people who were there who was like, "There's no snakes here," and then, "Oh yeah, let me tell you a story about how that happened," and then that was a myth, and then people had snakes as pets, and they got loose, and now.
Right.
Yeah.
So it has a serpent-free reputation, but people own lots of snakes, and there are wild escaped snakes. But according to mentalfloss.com,
Which is a very trustable.
very reputable site, as of yet,
Not
no
bad.
species has managed to take hold in the wild.
Okay.
So yes, and then the other thing that I found was 3 million years ago, when Ireland left mainland Europe,
Yes.
there were obviously many snakes. But what happened was, when the Ice Age came, the cold-blooded critters died out, and they have not since been able to get back.
Oh,
They haven't been
interesting.
able to take over ecosystems.
Yeah.
Look how good a show we do when we just wing it.
Just go. Let's just ask questions and answer them. Let's figure them out.
Part of being a scientist is like being a really good crunch time
Google it.
fact checker, right?
Being able to
That's
Google
right.
stuff when you
Is
forget.
it a myth? Is it true? Let's do this. All right, good. Did anyone save a spider this weekend?
I didn't.
Well,
kill
Blair,
a
I'm not even
spider.
asking you.
I didn't kill any spiders. I know you're trying to avoid that. Anyway.
I definitely didn't not save any spiders this weekend.
It's like, come on, spider, I'm going to rescue you. No, this is my home. I live here. No, come on, I'm going to get you out of. No. Please leave me alone. I'm fine right where I am.
As long as a spider is not, like, I don't know, near the head of my bed or in my food, I'm happy with them living in the home because they're getting rid of other yucky things.
Okay, well, this weekend was Save a Spider Day on Sunday. So if you did save a spider on Sunday, congrats to you. That was also in the process of losing an hour of sleep for daylight savings time, which is still very contested, as you will see in many media articles and across social media this week. People fighting for standard time, other people fighting for daylight savings time. I don't know. I think if we could just pick one, it would be a lot easier.
Just pick one. Why are we moving around? Why are we messing with our circadian rhythms?
Well, it's the circadian rhythm stuff that's really the issue, isn't it? Yeah.
Is it even? Like, it's not a big deal. Just deal with it.
I'm so tired. Still.
And on this St. Patrick's Day, Pie Day. Did anyone get to eat a pie? We missed Pie Day on Sunday.
Yeah,
I almost didn't eat a pie. But one of my friends magically, mysteriously had a pie delivered to my front door.
Oh my! Oh, that's great. That's like you mixed Sneak a Zucchini Day, which is also on the calendar.
It was Sneak a Pie Day. It was awesome.
Sneak a Pie Day. That's great.
Thank you, JoJo and Heather. All right, so let's get to the science. Enough with all of these special dates. March is big. There's a lot of stuff going on. We have great stories. I've got stories about Irish eyes and eyes of all kinds, really. And I've also got no womb.
I'm sorry. What?
Well, a story.
No room?
No womb.
Oh,
no
okay.
womb. We'll talk about that in a few. Justin, what did you bring?
I have got Sunken Treasure of Cosmic Design, Bad Air that is really old, Fast Food, go figure why oceans are worth protecting, and The Green Acres of the Arctic.
Ooh, I was going to start singing the Green Acres where I used to be. Greenland in the Arctic, I see. Okay, Blair, the Animal Corner.
Yes, the Animal Corner tonight is for the birds. I have a story about birds that can't mate, and I have a story about birds that can't parent. So some birds with some trouble today. And then also for the quick stories at the very beginning, I have a story about why masks are bad. I mean, they're good. We know that they're good, but they're also bad. We're going to talk about it.
Okay, fantastic. As we jump into all of the science, I would like to remind everyone that if you have not yet subscribed to This Week in Science, you can subscribe to us on the YouTubes, on the Facebooks, on Twitch. You can also find us all podcast players that are out there. I'm pretty sure pretty much all of them. Look for This Week in Science. You can also head to our website, TWIS.org. All right, ready for some science? No womb for the science.
Oh.
And maybe it's not a problem. Maybe it's not a problem if there's no womb. Huh?
What? Is that what you're waiting for? What are you talking about? Why do you keep saying the word womb?
We need wombs. Wombs are part of the deal for...
But what if they weren't needed necessarily?
No. That's not.
Or maybe a synthetic womb instead. Researchers just published in Nature their work on mouse embryos, keeping alive little mouse embryos for a couple of weeks, actually. Initially, they were trying to grow embryos in a nice embryonic fluid bath, and they were able to keep them alive for about 3 or 4 days. But but then the cellular development didn't go much further, and the embryos ended up dying. And the researchers played around with a bunch of ideas and eventually realized that they needed to increase the air pressure and oxygen delivery. And so they created a mechanism through which they could deliver the needed oxygen nutrients to these growing baby mice. And they were able then to keep the mice alive for up to 11 days to a much later state of development. You know, it's not fancy. It's a bunch of lab equipment, really, and modified centrifuges, modified pumps, moving fluid around, stirring little balls of cells, blastocysts, the hollow ball of cells, the gastrula, and taking them to a point where they actually started to grow organs inside of the cells. They differentiated and began to grow organs. This is the first time this has been done. It potentially, for lab research situations, opens up, according to a developmental biologist on the Science magazine website, Magdalena Zernicka-Goetz, it opens new doors by making embryos accessible for a detailed study of many aspects of their development. So being able to not just culture cells up to a certain state, but actually get to the next stages of development without having to go into the pregnant womb, remove these cells, and do that aspect of study. It's going to be a lot easier to push the process of research forward.
How is this different from lamb bags?
Yeah, so that's a great question. So it's the opposite direction. Lamb bags were for, or are for, what we've reported on before, these synthetic wombs that in effect are great for late term births. So, or early term births. So when premature births happen, these lamb bags could be a nurturing nutrient environment. I mean, right now they're called lamb bags because they're being studied using lambs. But the idea is that they would be able to give that womb-like experience to a premature baby that can't survive on its own, or that needs a little bit more support for just a little bit longer. And it could, so the lamb bags are coming from the end of pregnancy, kind of moving backwards. And this situation is starting from those fertilized cells at the very beginning of development and moving forward. So somewhere in the middle, maybe the two technologies will merge. Yeah.
So this is the other thing I'm wondering about this. Is this something that you could use to develop an extinct species embryo? Because
Probably.
Generally you find a good vessel, right? So you're like, if I want to make a mammoth, I'm going to put this mammoth embryo in an elephant.
And we definitely do.
And in this case, the vessel is a jar.
Right. But if it was a mammoth, it'd be like a really big jar.
Well, not at first. It would just have to be transferred
Eventually.
To become
It'd be like a potted plant.
Successively larger jars, and then a lamb bag, and then a mammoth bag.
Right. Nice.
Yeah, so it's pretty interesting. This could potentially be a direction for also for in vitro fertilization, for the to really make sure that the development of embryos is progressing in the way that you want it to progress before implantation, before actually transferring them to a mother's womb. There are a lot of different reasons this could be very interesting. I mean, in the sci-fi take on this, at some point, like you brought up the lamb bag, and there's this, at some point it will lead to embryos being taken to full development in some way. For sure. It'll go there.
No. Can you imagine if this technology was available this moment?
Yeah.
I want to have kids, but I don't want to get pregnant and go through all that nonsense. So I'll just hire this lab.
Right.
I'll pick up my baby when they say it's ready.
Yeah. And then you wonder, like, all of
I don't know
the
about that.
language and the heartbeat and that
Yeah.
connection stuff. Maybe it turns out doesn't matter at all. Never mattered, actually. Just people like to think it mattered. Didn't matter at all. Could be. Or it could be very important, and we just had no idea how important.
Or you carry your jar of baby around with you like the sack of flour in high school.
No. No. Make sure to shake your baby once a day.
Never shake a baby.
Talk
Never.
to your
Just-
baby in a jar.
The
Hello.
more you know, don't shake baby.
Tell that jar
Ever.
baby you love it.
How
Oh
is
my
it in
God.
there, little baby? Swish, swish, swish.
And additionally, like tie on to this
Child's
news.
first words: never knock on the glass.
Oh no.
Don't ever do that again.
And a kind of carry on to this is another study that was also, or a couple of studies that were published in Nature this week, and there were some preprints in this vein as well, related to the study of human fertility. It's been very difficult to there are limits on studying human blastocysts, on studying human cells for ethical reasons, and there have been choices made and regulations taken so that the study of human cellular development is limited in many places around the world. However, researchers at the University of Texas Southwestern Medical Center, they have taken skin cells and they were able to get them to become embryonic stem cells and then pretty much get them with chemicals to turn into blastocysts, which are the first stage of organism's development. There's fertilization. The egg then begins dividing, and then you get a little tiny ball of cells that's the blastocyst. After the blastocyst, you have this invagination that turns into the gastrula, which is that ball of cells that has, it's like a donut, basically. But what they're doing, they've created these human blastoids, basically these balls of cells that are able to grow in dishes in the lab that did not come from fertilized egg.
Right.
They didn't come from that. They came
No.
backwards. It was like,
No,
here's
but-
my skin, and now I'll make some things that are very close to what the
Is
developmental process is.
that basically a cloning technique then?
Kind of, yeah. I mean, the question is, they have the
Does it
potential.
become viable?
That is the question. Yeah. Could it work that way? I mean, we've got blastocysts. If you then put them into the mouse jar, the baby jar, can you get
combine the
them to
two.
grow and then to the stage of the lamb bag? Can we get that to work? I don't know.
And then you're like, I think this person has been very intelligent, very successful in their life. Hey, can I shake hands with you? Thank you. Swaps up. What are you doing? Oh, nothing. Just want to have successful offspring.
Okay, so
So strange.
the researchers state very carefully that these blastoids are not embryos. So they're not going to undergo those early stages of embryogenesis. I mean,
But
number one, they're not
they're
getting the right
growing.
they're not getting the right molecular signals. They don't have the right conversations going on at the chemical molecular level.
But they're growing.
But they are growing. They're cells that are growing
What are they-
together. Yeah.
What are they growing up to be?
Yeah. But all countries, a
big-
bunch of countries have
Just one.
decided that human blastoids, cannot be allowed to grow for more than 14 days. Because after 14 days, the question of their viability as a developing human, I think, becomes more questionable.
But
I
how
do
else are we
agree.
going to know if it could?
Nah, so it's annoying. Yes, there does need to be a cutoff. I think at some point we will all agree. But should it be 14 days? That seems early. But
Yeah.
then the argument can go on forever there.
Yeah. I don't know. I mean, though these blastoids that are being developed right now, no, they're not going to turn into embryos. They're not going to be able to turn into babies. But this is the kind of technology that leads us in the understanding that will allow this kind of stuff to happen in the future. So these studies that are out today are steps in the path of technologies related to in vitro fertilization, understanding issues with fertility, of being able to foster children outside of a womb, given if there are medical conditions that won't allow you to have your own child. There are all sorts of things that there are use cases, and the technologies are developing. Very interesting to me. All right, Justin, tell me about a shipwreck.
All right. Okay.
Let's move forward.
Wait. You're getting ahead of the story. You told us. Okay.
That's the headline.
Oh, yeah, that's right. Hoist your sails when the winds are fair and the seas are smooth, but never mistake smooth sailing for skilled sailors, because bigger ships with better crews than the one you are in have sunk beneath these waters. And on one such shipwreck, lost to the bottom of the Mediterranean Sea nearly 2,000 years ago, discovered by sponge divers 120 years ago near the Greek island of Antikythera, was a strange lump of corroded bronze and wood that, upon closer inspection, had a gear of some sort embedded in it. Mystery gear turned out to be part of a larger mechanism, which eventually revealed it operated as a mechanical clockwork calendar with 30 gears and many inscribed discs and even some tiny beads that played a role somehow. It had the 12 zodiac symbols, Greek writing, the Egyptian calendar, days of the year. They were kind of interesting. We were talking about they had 12 30-day months, and then 5 days that you got through and you forgot the evening. Did those 5 days, but the rest of the months were all the same.
Sounds like a 5-day vacation for everybody.
It kind of was like this time doesn't count, but we got to do it. If your birthday was on this day, sorry, you don't exist. Researchers at the University College London have discovered that it could do even more. So they found that it can show the ancient Greek order of the universe, which is basically what we call the solar system today. The planets, I think they had 5 planets at that time. This is quoting the lead author, Professor Tony Freeth of the University College London Mechanical Engineering Planning. Ours is the first model that conforms to all the physical evidence, conforms to all the physical evidence, and matches the descriptions of the scientific inscriptions engraved in the mechanism itself. The sun, moon, and planets are displayed in an impressive tour de force of ancient Greek brilliance. So they've kind of worked somewhat off of some past studies. 2005, there was 3D X-rays and surface imaging enabled them to show how the mechanism predicted the eclipses, and it calculated the variable motion of the moon. And it uncovered, too, that there was thousands of text, thousands of icons of text within the mechanism. Let's see. So about a third of the mechanism has actually survived, so they're missing most of it, and that was split into 82 fragments, so there was a big puzzle to put together in the first place. Biggest surviving fragment is known as Fragment A. It displays those features bearing pillars and a block. Another is Fragment D, which has featured an hereto unexplained 63-tooth gear and plate. This is pretty complicated mechanism. They found thousands of text characters hidden inside the fragments when they did the X-ray stuff. They found planets moving on rings and indicated by marker beads. And it was this that the team was trying to reconstruct. And one of the things that the X-rays showed from the front cover was something that said 462 years and another that said 442 years. Sort of interesting, because that happens to accurately represent the cycles of Venus and Saturn, respectively. They actually had calculated the motions of Venus and Saturn. When observed from Earth, the planets have cycles that kind of reverse themselves. They didn't have it like an elliptical. It would like go way over here and it's going to move back again at some point. So they had to track all these variables over these long periods of time to be able to predict their positions, and they did that through the mechanism. So they still didn't figure. Okay, so this is quoting voice from, remember, Eris Dickanalis of the research team. The classic astronomy of the first millennium BC originated in Babylon, but nothing in this astronomy suggested how the Greeks found the highly accurate 462-year cycle for Venus and 442-year cycle for Saturn. So using some ancient Greek mathematical methods that they had found had been described by the philosopher Parmenides, the team not only could explain how the cycles for Venus and Saturn were derived using that ancient math, but also managed to recover the cycles of all the other planets that were missing, and then be able to find them in the mechanism. In fact, this is David Higgin, who's a PhD candidate member of the team. After considerable struggle, we managed to match the evidence in fragments A and D to a mechanism for Venus, and as well that's completely related to that 63-tooth gear that was playing a crucial role. The team then created innovative mechanisms for all of the planets that would calculate the new advanced astronomical cycles and minimize the number of gears in the whole system so that they could fit it into these tight spaces. They still have to go back and they still haven't figured out how to truly, fully reconstruct this thing, partly because there's a bunch of things missing. But there's also these little tubes inside, and these beads will apparently, from the way it sounds, the beads will sort of drop and show like, oh, this planet is now going to be here, and we should look for this one here at certain occasions. So it's insanely complicated mechanism, 2,000 years old. One of the first. Wait, the surprise part of this. Somebody's asking, wait till Justin gets to the surprise part of this. I thought that was the surprise. That's the surprise. They could calculate the planets with.
So they had a calendar that was not just a calendar, but a solar system, not a solar calendar, but a solar system calendar.
Solar system calendar. And maybe this is the surprise part. And it could also tell you when the next Olympics was about to happen.
Of course, because you need to be there to start running at the right time. Yeah. You got to get there at the right time. So did they think that this was a common device, or was this something that was like a specialty item that only certain people, was a one-off?
So it's a one-off as far as history is concerned.
As far as history is
anything
concerned,
like that.
Yeah.
Okay. As far as even a description of this is missing.
The question is, did the person who had it, did everyone think that they were crazy?
So it was in the shipwreck. They think it might have been looted from somewhere and was being transferred. It was on a Roman ship. It's got Greek lettering, and it's tracking Greek stuff. Egyptian calendar, but it's a Greek thing heading to Rome. It may have been looted from a military campaign. The thing is, we just don't. We just don't know. There's been nothing else like it or hinting at it ever.
I just picture someone saying, I know where the planets will be. And everyone's like, yeah, all right, Tom.
Yeah.
There he goes again.
It's pretty incredible.
Put your weird box away.
I say that there's been nothing like it found, and that's pretty much true. But there have been accounts of other mechanized, sort of robotic, animatronic devices and things
that go way back.
But have not been found.
Yeah.
And then it's like, oh, that's just storytelling. Then you find a thing like this and go, wow, they had a very complicated understanding, somebody did, of clockwork gearing 2,000 years ago.
That's what impresses me is the number of gears, the complexity of the sizes of gears, the way they interlock with each other, the way they move together. I mean, that in itself is mind-boggling to consider the complexity of how they all work together. But I'm not a clockmaker. I've never tried. I could probably deal with a couple of gears but you start putting more together and it raises the levels of complexity.
How big was this thing, Justin?
Oh, that's a good question. There it is. Is that no, that's not. Is that centimeters that it's showing there?
Oh,
No.
the
No, that's
centimeters for some of the gears.
Oh yeah, like 10
individual
centimeters.
gears. Yeah.
Yeah.
Which, when you think of making those gears.
That's inside the
that's-
larger mechanism. Yeah.
Yeah.
Hmm.
Pretty cool.
Mhm.
This thing we've been talking about this mechanism, the Antikythera mechanism, this mystery for decades. People have been looking into this, trying to, what is it? What secrets of the universe does it hold? It's pretty awesome with modern technology and some puzzle solvers, pretty much. These scientists who've been working so hard to figure out how it all pieced together and figure out the puzzle.
And there's still thousands of icons that we haven't really figured out how they play in. I mean, this is also part of the incredible nature of it. It's like, what if it does something else?
This is amazing so far.
Yeah, it's pretty cool,
impressive. Yeah. All right, Blair, you have a story for us?
Yes, about masks.
Are you going to badmouth the masks?
I am and I'm not. So, so of course, at the beginning of the pandemic, we were saying, don't wear masks, save them for the medical heroes. And then we said, wear them to protect everyone else. And now it turns out all along, they were protecting us too, right? So they're very essential to our safely getting to the other side of this pandemic. And I am not going to say anything to the contrary because masks are essential to that. But let me ask you this. What is a surgical mask made out of? Do either of you know? Have you thought about it?
Plastic fibers.
Mm-hmm.
It's not made of nice cotton, is it?
No, it's not cotton. It's not paper. It's plastic. These disposable face masks are made from plastic microfibers. And you can see where this is going. It's an ecological nightmare. So this is a problem because, of course, right now we are throwing away around 129 billion face masks every month. That's 3 million a minute. But even after the pandemic is over, these are needed for the medical field. So it's not like it goes away overnight, right? Disposable masks are here to stay. And this is a big problem with the medical field in general, right? There's a lot of disposable things. There's things you can't use over and over. There's things that you can't sterilize. There's things that it's safer for everyone if you have a disposable product. But when you look at these disposable masks that are made out of plastic, it can't be biodegraded, but it does break up into small plastic particles. They're micro and nanoplastics, and they are already widespread in ecosystems. There's no official guidance on mask recycling. Plastic bottles, for example, you can recycle, but you can't do that with masks right now. So they are disposed of as solid waste. Even the ones that make it into the trash, which we know it's been a huge problem of people for some reason just taking the mask off and throwing it on the ground. I don't understand what's going on there.
But
ultimately,
like all the other things.
Even if you're putting it in the garbage, a huge amount of it will end up in the water column anyway. And so these masks that are made out of micro-sized plastic fibers, they have a thickness of about 1 to 10 micrometers. When they break down in the environment, they release more micro-sized plastics that are easier and faster than bulk plastics like plastic bags to move around and get digested and all sorts of other things. There's actually a new generation of disposable mask coming out called nano masks. You might think that might be better. Ooh, no, made out of nano-sized plastic fibers.
Even smaller.
Yeah, the diameter of smaller than 1 micrometer. And they also are probably going to be a new source of nanoplastic pollution. So the other problem is, I didn't even think about this. They have buildup of harmful chemicals if you're wearing them to work with chemicals. They have buildup of biological substances like bisphenol A. They have heavy metals in them. And if you're using it to filter out microorganisms like pathogens, then those end up in the water column. So that's all part of this kind of problematic situation with the masks. So what can you do? The researchers who are kind of looking into this, there's no official research yet on the microplastic, kind of the depth and breadth of the microplastics from disposable masks yet. That's in process. But the people who are looking into this have 4 suggestions for disposable masks. 1, set up mask-only trash cans for collection and disposal. Makes sense. Then they're not going to fly out of your kitchen trash, right?
Yeah.
2, consider standardization guidelines and strict implementation of waste management for mask waste. So that could be a way to maybe recycle them or find a way to seclude this stuff in a safer space. 3, replace disposable masks with reusable face masks whenever possible, like cotton masks, which a lot of us are doing, but that needs to be kind of pushed forward whenever possible.
And if you're using the cotton masks, make sure that you're double masking, because that's why they say double mask now. It's not that, oh, you're wearing an N95, double mask with N95s. No. It's just put a couple of cloth masks on to really make the filtration better.
Mm-hmm. Absolutely. And the 4th suggestion here is to, of course, consider development of biodegradable disposable masks. It's got to be out there. There has to be a possibility for this. So that's actually the main reason I brought this story, is that a lot of people probably thought their surgical mask was made out of cotton or paper, and it's not. It's made out of plastic. And somebody might have a really great idea for how to make a biodegradable mask made out of potato or corn. Wouldn't that be great?
Well, I mean, a lot of plastic does come from biological sources. We do have plastics that come from potato, from soybeans, from, bamboo floors, bamboo polyester comes from bamboo, so there are natural sources to the plastic fibers. It's just that they are a certain conformation of molecules that leads to the way that they work within the environment and how they don't break down as easily. I'm thinking, though, the thing that they're missing here is using plastic-eating bacteria. Like, we should get some kind of, you get your plastic-eating bacteria glop, and you slop it into your bag of disposed of face masks, and then throw the whole thing away.
Yeah. And, when there's a global pandemic on, maybe you have your mask bin and you put out mask waste once a month, and your waste management company picks that up, and that goes into a giant plastic eating glop.
It would be hazardous. It would be, quote, kind of hazardous waste disposal at that point, depending on, you don't know what has been breathed where or how. And so that would be an interesting, but a waste disposal company could deal with that. But simple to compost.
Medical and labs do that already. They have, biological hazard bins that stuff goes into. The specific crew comes and collects and takes away. What they do with it, I don't know. Probably maybe it just goes to the dump, but they at least transport it.
It goes to a special dump.
Yes, a magical special lab and medical dump.
I like the idea of, you've got near your big grocery stores, sometimes you have like the bottle drop, and you've got the plastic recycling, and you can go take things, and you've also got your face mask disposal. Just everything in the same place.
I mean, I wear disposable contact lenses, and so I collect all of the contact lens cases and the contact lenses themselves. I have a special little disposal for them, and those get dropped off at my optician, and she sends it back to the company, and some of it gets recycled, and it prevents those little tiny microplastics that are in disposable contact lenses from ending up just in normal trash and in the water column. Yeah.
So, how do you prevent cancerous cells from spreading?
Why don't you tell me?
Yeah. Counter to some, I don't know, woo conspiracy theory ideas that you might hear out there, some researchers at Ohio State have discovered that electromagnetic fields could slow or stop certain kinds of cancers. When cancerous cells were exposed to electromagnetic fields in the laboratory, they found that it inhibited the cancer cells' metabolism, did not inhibit normal cells, but it stopped cancerous cells from expanding and traveling and growing in the way that cancerous cells like to grow and develop. The researchers think that it selectively works by changing the electrical fields inside these cells, that somehow accesses the internal workings of the cell. But this is something that is a kind of new discovery based on their work, and so they have more work to do for sure. It started more questions.
Okay, so one of the things about cancerous cells is they have stopped listening to all the neighboring cells. They've stopped listening to the orders to stop growing. They've stopped listening to the apoptosis orders. They stopped listening to anything other than we're in go mode. Cut all the communication to the outside world. We're just gonna grow.
Go, yeah.
So, some sort of disruptive, you might call it, information comes through from this magnetic field. Gets through. A normal cell, it's like, okay, what's that? That's noise. Because we're all in communication. That's noise. Don't need it. It gets into that cancer cell, and the cancer cell is like, oh, wait, we got a signal. Do we have orders? We have new orders. Do something. Wait, we just gotta figure out what it is. Like, something finally gets through. So there's a way that kind of, like, just
makes sense to you in a way.
That it could be. But it does also make me laugh, because then all these people who are trying to get away from, like, high power lines and, like, being afraid of the meter on the side of the house, because that causes cancer. Actually, just like when they took the mercury out of the vaccines and the autism rates went up, apparently autism was preventing autism. That's not how that works. But you're making up pseudoscience, health fear remedy junk. It's actually killing you.
Yeah, no, I was I was thinking about this. You know, it's like all these people who are upset about the electromagnetic fields around high power lines, about cell phones, about all these things. And it's like, wow, if this study has something to do with it. And to me, this is a very interesting opposite, completely counter to what they're positing kind of result. So.
The Ötzi the Iceman? How old is Ötzi the Iceman? Is he a 3,000-year-old?
I don't remember. Yeah. 3 to 5? Yeah.
High cholesterol. Yeah. He had has nothing to do with the modern diet. He was like a 40-year-old man with high cholesterol. It was even normal then.
Yeah.
That's just how humans have been for a long time.
Yep.
I just didn't have a word for it.
All right. Anyway, I kind of feel like maybe if this work goes further, we'll keep an eye on it and see how these electromagnetic fields end up actually tying into why they affect cancer cells in this way. But I don't know. Maybe there's going to people be using electromagnetic fields as like a cancer force field.
They're going to be using that way ahead of any research and development. This is the problem with information. Like sometimes it's just better.
A little bit is too much.
Shh. We just found this thing, but we're going to be really quiet about it until we can get more causation involved.
Yeah.
Somebody's going to totally misinterpret it and start kicking around a microwave. No!
What's up? I'm just wearing my magnet shirt. It's just full of magnets. But I apparently I went to go get in the car. I think I have too strong of magnets. I got up to the door and now I'm stuck here. How do you turn off a magnet? It's really difficult.
All right, tell me about some bad air.
Okay, so the Roman word for air is area. Their word for bad was mal. Put them together and you get area mal, which is full of suspect air that might be bad these days. Put them together the other way around and you get malaria. Romans believed that bad air of some regions were responsible for the disease that they called malaria. Long thought to be a disease first encountered during the birth of human agriculture when we started grouping together and irrigating and then being around standing water. There would be mosquitoes. We think this is how malaria first started to interact with humans. But now new bioarchaeological research shows that malaria has threatened human communities for more than 7,000 years. This is lead author Dr. Melandri Vlok, Department of Anatomy, University of Otago, says this groundbreaking research published in Scientific Reports changes our understanding of how long humans have been dealing with malaria, which, by the way, still one of the deadliest diseases in the world. 2019, World Health Organization estimated 229 million cases of malaria around the world. And if you get malaria and you die from it, most of the time you are under the age of 5 years old. This is a very brutal, lethal ailment. Research has identified thalassemia, which is a thing that you can get. It's a genetic disorder that causes porous bones, can have some very devastating effects. However, for some reason, it also offers, in the milder form, some protection against malaria. Researchers identified thalassemia in an ancient hunter-gatherer archaeological site from Vietnam, dated to approximately 7,000 years ago. This is thousands of years before we had the Green Acres farming stuff going on. And in this particular region, mosquitoes are common in the forest. So you don't have to have that irrigation, pools of standing water scenario for that high degree of mosquito and human interaction. It was they're present there in the forest. So pretty interesting. They had used microscopic techniques to investigate changes in bone tissue to identify the disease. They had found it in a 7,000-year-old hunter-gatherer site, and then they found it again in a 4,000-year-old agricultural site in the same region. So then you have two examples separated by 3,000 years. This is Dr. Vlok, quotey voice. A lot of pieces came together. Then there was a startling moment of realization that malaria was present and problematic for these people all those years ago, and a lot earlier than we've known about until now. So, just a little bit of advice from the past.
Right.
7,000 years ago. Diseases have a way of sticking around.
Well, and so, you were talking about how devastating this disease is, but that it kind of, how do I put this the right way? It's like when we've talked about on the show how an effective pathogen doesn't have a 100% death rate because it has to stick around, right?
Yeah.
And so you can see how if malaria is stuck with us for this long, it's done, in its view, a really good job of being deadly enough, but not too deadly within our population. And that might be why it's stuck around so effectively for so long.
And it's still and it doesn't affect everybody as badly. And I guess if you have access to medical, it's not a big deal. A friend of mine from back in the day, who's a physicist from India, told me he'd gotten malaria like a number of times. Like he was like, oh, malaria. Yeah, I've had that many times.
I grew up with it.
Yeah, it was just common in the region he grew up in that people got malaria. That's just the thing that happened. It's like this seasonal malaria came along. Seasonal flu here, seasonal malaria there.
Yeah, it's like, oh, Joey's not in school today. Oh, yeah, he has malaria. Like, oh, God.
We know that a lot of diseases, a lot of viruses, a lot of parasites have stuck with us for thousands of years, hundreds of thousands of years. I mean, there's been research into the herpes virus, which goes back as long as humanity has been around and even further. Like, it's been there are diseases that have evolved with us, that stick with us. There are ideas that previous COVID viruses that are now common flus caused major pandemics in the past. And then they eventually, lots of people died and then turned into a common cold because herd immunity and and also possibly changes in the virus enough to make coexistence possible. Yeah. So malaria is another one. Been around. It's been
at least 7,000.
At least. Probably longer.
Probably much longer. Yeah.
Mm-hmm. Yep.
Because Southeast Asia, that's a pretty early, one of the earlier sites that
Yep.
reached. And then there was Denisovans were already there.
Mm-hmm.
Maybe like, did they have malaria issues? Like how far back does this
Why
go?
not? Were there mosquitoes? Were there biting insects? Were there human-like organisms? Yes. Yes. Most likely. Okay. Blair, what's a giraffe's blood pressure like?
Well, it has to be. That's kind of a funny question. So it has to be really high because it has to pump it up to its head, which is why giraffes don't lay their head down for very long ever. It's because they'll get just the worst head rush. So this giant, powerful heart, and if they put their head below their heart, they can black out.
Yeah, it's like their I guess equivalent of standing with their knees locked, right? All the blood stays in their head.
What happened? Oh, I kneeled down too quick.
Well, researchers decided to dig a little deeper into the giraffe genome to find out more about the genes that allow the great blood pressure and height and behavioral adaptations of giraffes to take place. So they looked very deeply at the chromosome level giraffe genome and compared it with other ruminant genomes. They looked at a bunch of giraffe-specific mutations and found a lot of mutations in a gene called FGFRL1. And this is a gene related to blood pressure. So what did the researchers do? They used CRISPR to put the giraffe gene into mice to see what happened to the mouse ability to deal with high blood pressure when they had the giraffe genes. So basically, they took giraffe genes, put them into mice, and then gave the mice high blood pressure and checked to see what happens. And the mice were like, dude, high blood pressure? That's fine. I can totally deal. No problem. Yeah. Yes. So these mutations, not just the giant heart of the giraffe, but also genetic mutations have allowed giraffes to actually manage constant high blood pressure. And we might be able to use their mutations or understanding of how those mutations work to help human high blood pressure. Maybe we can find genomic level treatments for people who are have very high blood pressure at some point. I mean, or maybe there are targeted treatments that can be targeted at certain receptors based on what we understand about this at some point.
I just pictured, I got a pig heart valve and giraffe genes.
That's right.
I immediately pictured
Human zoo.
Picture a conference. It's maybe 50,000, 100,000 years into the future. It's a conference. And as the lights go up at the conference, you realize that somebody's at the podium about to speak, and it's all mice. And the mouse is announcing, we have discovered the cure to high blood pressure from ancient ape texts that we have deciphered, curing yet another disease that has malaffected mice kind for so long.
Yeah, eventually we'll all just have immortal mice, right? Is that the deal?
They're just like, they'll be like these ancient apes. They spent so much time curing all of mouse-kind's diseases.
And giving them diseases to mice. Oh my goodness. Another thing that they found is that the researchers found genetically edited mice did not have so they didn't have this normal reaction to this compound Ang2, which is an angiotensin, high blood pressure related gene. They additionally noted a sensory trade-off in giraffes. So the genes of their sense of smell were reduced and downgraded, and vision genes were upgraded. So there's more more stuff going on in giraffe vision than there is in giraffe smell. And the researchers hypothesize that this is because giraffes rely on horizon scanning vision from the tall vantage point, and that olfaction at 5 meters from the ground probably isn't as important as it might be for other animals.
Well, I mean, and they're not working too hard to sniff out food. I don't think acacia smells too much. Just leaves.
Yeah, if you think about how they forage and what they're doing, right? Oh, I see some over there, Bob, as opposed to, I smell the wafting of acacia leaves on the breeze.
Mm-hmm.
Yes, no. But anyway, yeah, big discoveries thanks to giraffes could help high blood pressure. Giraffes and mice, giraffe-mouse combos. Anyway, Justin, did you have another story you want to talk about right now?
Oh, I did. I have a really fast story. This is the quick story. Researchers in Australia have found that for each new fast food outlet in a region, the number of heart attacks per 100,000 people goes up by 4. Team found that the fast food was positively correlated with an increase of myocardial infarction, even after accounting for other factors such as age, obesity, high cholesterol, hypertension, smoking, diabetes, associated economic status. All of these things were factored in. And while correlation isn't causation, it is a tool used to discover causation.
Absolutely. It is. It's a step in the direction of discovery.
But that's the fast story of the week.
You know
fast
what that
food.
is? That's the French fry factor.
You think you're blaming the French fries straight off? Okay.
I am. I'm going to blame the French fries. French fry factor. At least I'm not going to blame them. I'm just coining the term right now. Okay. You heard it here first. This is This Week in Science. If you just tuned in, we're talking about all the science from the week. Are you enjoying the show? Please tell a friend. This Week in Science. Okay, I'm going to come right back. And hey, can we have a little bit of a COVID update?
Yeah, I think it's time. It's been a bit.
Time. Okay, let's have a bit of a COVID update. COVID update. Whee! Well... Okay, in vaccine news, AstraZeneca, that is the Oxford vaccine, which has not yet been approved here in the United States, although it is under review, has been put on hold in several countries after reports of blood clots following administration. Many of the reports, there are conflicting reports in that some reports say that the numbers of blood clots are not more than would be found in the average population without the vaccine.
Right.
And others say that the numbers of blood clots are higher than would be found on average and are within a certain time frame, a short time frame after getting the vaccine, and so are indicative of a cause. However, it's still contested. There's a review that definitely has to take place. But in many places, they're talking about potentially just stopping the use of certain batches with the question of whether certain batches, because the AstraZeneca vaccine is manufactured in multiple plants in many different places, that certain batches could be bad or having these effects for some reason. There could be differences in how they're being manufactured. There's a lot of questions at the moment. But to put it in perspective, the numbers of blood clot effects are still much, 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 much less than what you would actually expect your chances of getting the virus to be.
Mm-hmm. Yeah, that's what I was trying to find. I want to say it was like, this can't be right. But I thought it was like 40
really
people.
Out of like 50 million vaccinations. Yeah. Yeah,
maybe,
so
So
yeah. So the numbers, if you remember, there was a brief pause on the Pfizer vaccine when it first was let out, and because in the UK because of the question of allergic reactions, and now we know there are some people who do have allergic reactions, and so there's a period of like, okay, let's watch you to make sure you don't have an allergic reaction, and let's keep an eye on that. And that's taken into consideration. But there, even though it's not happening to a lot of people, this is part of the process. You want to keep an eye on it and stop something before it does become a lot of people. We don't want any of these treatments to become worse than the actual disease.
Well, and you would hope that out of these 40 people or whatever it is, it's such a small number, it'd be hard to even point to anything specific. But if there was something in common with these people, like for example, we were just talking about cholesterol, like maybe high cholesterol had something to do with it or something like that, then you could select for that and say, maybe you're not a good candidate for this vaccine.
Right. And we don't know that yet. So perhaps that will become understood and that news will be passed along at some point. Yeah. But in that as well, the AstraZeneca vaccine is also not working against the B.1.351 variant of COVID-19, which is the South African variant, which is a special vaccine escape concern. So apparently a new paper out in New England Journal of Medicine, AstraZeneca doesn't work against that particular variant. So if you get the AstraZeneca vaccination, there's a limit to how much it may protect you against future variants. And you may be in the next few months, year, looking at definitely getting a booster shot of something else. What were you going to say, Justin?
Yeah. Oh, well, so one of the confounding elements of this, and I get it's been, great minds are at work trying to sort this out right now.
Yeah.
But one of the interesting things is there was a correlation of people who had gotten COVID and survived it, who then had strokes. So I think part of it is just we're looking at very large populations and all of this, but it's so hard to separate the noise
from a signal.
Yeah.
Mm-hmm.
Except that I guess it was specifically like not linked to high cholesterol and this sort of thing. So these were outliers, which is why it got the attention that it has garnered.
Yeah. And then speaking of variants, the UK variant B.1.1.7, new research, says that B.1.1.7 is not only more transmissible, but up to 68% deadlier. Now, with 68% more death, as compared to other circulating COVID variants. But the good news with B.1.1.7 is that as far as we are aware, based on current testing, it does appear to be neutralized. There's a slight reduction in neutralization effectiveness, but it does appear to be neutralized by most of the vaccines that we are currently administrating. So that's good.
So there's a couple things I want to point out about this that I think is really important. Is for somebody just kind of reading this headline, they could see 68% deadlier and turn that into 68% deadly in their head. And that is very different.
Very different.
Very different, because it's 68% deadlier than the virus that we've been tracking that's like 1.7% or something like that. So it's higher, but it's still it's a smaller number. So I don't want people totally panicking over the variant. But the other thing I really want to point out is this is one of the reasons why even if you got a vaccine, you're still supposed to be wearing a mask when you go out in public. And this is exactly why, is that yes, the vaccines still cover by and large the variants. However, the only way for us to get past these deadlier variants is to reduce spread. And the way to do that is by masking, even if you've been vaccinated, because you might be carrying that deadlier strain. And so being able to prevent that from happening is really important.
And one of the things that is being talked about more and more, and we've brought it up on the show here, is the difference between a vaccine that is prophylactic in treating the disease versus treating transmissibility. Mm-hmm. And So far, what we know is that these vaccines are fantastic at preventing disease. And so, like you've said, asymptomatic transmission can occur. However, there are researchers looking at viral loads in nasal swabs of people who have been vaccinated, trying to figure out how many people are infected and in fact able to spread the disease, spread the virus, even though they're not showing disease. And so a new study just came out looking at the mRNA viruses, looking at people who were vaccinated, and it's a broad swath of people. They looked at thousands of people to see whether or not they had were capable of, or whether they had asymptomatic infections after vaccination. And they found that the mRNA vaccines reduce asymptomatic infections by 80% after the second dose.
Okay, that's huge.
It's not 100%, it's 80%. So that's
huge.
Great, but there's still risk.
Yeah. And this is why the CDC said if you're around a bunch of other vaccinated people, you can hang out. Is because 80% and 80% and 80% and 80%. There's pretty good odds for you right there. But that's also why there's all these weird gray areas in the CDC's thing that they said, which is like, if there's one person who's not vaccinated from one household, then you're probably okay. But if there's two households of unvaccinated individuals, then you don't want to mix those because they're not protected. Then they can spread to each other. It's unfortunately, it's still very gray. And so you still have to be really careful.
Absolutely, but it's still fantastic news because the terrifying thing would be that everyone gets vaccinated and still carries and transmits and shares this virus. You're not getting the ill health effects now because everybody is vaccinated, and this, we're going forward maybe a year. I don't know how long this is going to take. Everybody's vaccinated, but the virus is now thriving in a population that doesn't die from it. And then a mutant finally comes along and we just jump right back into this again and none of the vaccines work. And one of the things about vaccines too is we've been talking about, there are still maybe a dozen or more vaccines that are being developed. The ones that we have now were emergency released.
Yep.
And their effectiveness is where it is, and that's great. And so we should all do it now to prevent the spread. But we maybe also then, again, a year, 2 years down the road, be getting vaccines that are 100% at everything. So we may in this arms race against this virus, we are now beginning to turn the tide, and we'll just, I think, be doing better in the future. Although I don't like predicting things because I also was like, this is probably just going to go away like the flu in the very beginning. Because it's novel. We don't know. It might just be. And I was really wrong then. So if I'm wrong again, as wrong now, oh, shoot. I should not predict things.
I think you're right on the fact that we are going to be dealing with some kind of vaccine for this virus for the foreseeable future. And even though you're getting vaccinated within the next 6 months with the emergency released vaccines, within another 6 months to a year after that, be ready to probably get vaccinated again. There will be more vaccines released. You'll probably want to get booster shots. There's the reason and the way that this arms race is going to work is with that development of new vaccines. Moderna is already testing a new target for a new booster shot for their vaccine. So the researchers are already on this. They know there are variants. They know what's happening, and they're trying to predict. They are trying to predict the future because they need to protect us in the future. But.
So in a really tragic, having to learn the hard way. Humans once again. Despite all of the warnings from science, despite people saying this, despite the bird flu, the MERS, the swine flu, but besides all these near events like this that we have managed to avoid, and then taking the eye off the ball and defunding the observers in the field, defunding the research into all of this stuff that takes place. Maybe it takes something like this to just, okay, now this is a thing we monitor and research constantly and have that as a true pipeline of funding. Do that research that does not get cut off or diminished because, hey, we haven't had a problem for a few years.
Oh,
maybe
hey,
this is what it's
all
at.
you capitalists who like the economy, you don't want this to happen again, right?
No.
How about we try?
Yeah. And I think it's worth mentioning, as long as we're talking COVID on the show right now, that, we just came across the year anniversary of the shutdown in the United States.
It has been a year.
And
we went past it.
Just in the last week was my anniversary of being sent home from work. And when it all started, we were saying, we can't wait for the vaccine. We can't wait for the vaccine. We have to figure this out. If we wait for the vaccine, 200,000 people will die.
535,000 people in the United States.
Yeah, so important to recognize, we thought we were being doomsday alarmists about it, trying to give this huge number, and it wasn't even half of what it was before the vaccine came out. And we said, we can't wait. We waited. It was more than twice what we thought. And so, I do think this is a really important reminder to stay vigilant and not let it get here again.
It's springtime. The schools are opening in my area. I know they've opened in lots of places, and it's springtime. I mean, luckily outdoors is much safer than indoors. So I hope everybody safely enjoys the outdoors as we move into our nicer months in the Northern Hemisphere. But it's still here. It's not gone yet, everyone, but let's keep moving. But you know what? We can have hope. I think that's the big thing. We can be optimistic. It's going to still take time, but things will start getting better. Oh, my goodness. All right. This is This Week in Science. Thank you so much for listening to us right now. If you are enjoying the show, please consider heading to TWIS.org and clicking the Patreon link. Patreon is how we are funded by you, our listeners. You're our supporters. You're the ones who help us continue to do this show every single week. We take a few breaks here and there, but really we're here every week with you. And we love to talk about science with you. So can you hang out with us and help us out and help us keep it going by supporting us on Patreon? Head over to twist.org and click that Patreon link. We really can't do this without you. Thank you for your support. All right, coming on back. It's that part of the show. Wait, what did you say, Blair? It's for the birds?
Yeah.
She loves our creatures, great and small. Biped, millipede, no pet at all. You want to hear about animals? She's your girl. Except for giant pandas and squirrels. And a whole
What you got, Blair?
Oh, I have the very sad story of why it is tough to be a regent honeyeater in Australia. So this is a study looking at regent honeyeaters over 5 years, tracking how they sing and how they learn their songs.
Oh, it is a bird. Okay.
Mm-hmm, it is a bird, the honeyeater. And their songs are how the males woo their females with their bird songs. Tale as old as time. And they usually learn those tunes from adult mentors. They leave the nest before they start singing when they're very young. So they learn their bird song from other adults nearby, not their parents. And so over this tracking of singing ability and breeding success in the critically endangered regent honeyeater, they found a very interesting change in how they learn their songs. They were common across Australia, but since about the 1950s, their population has consistently shrunk, and now there's about 300 to 400 of them left. Very small population.
Very, yeah.
On a large continent. And so they're spread out and they don't have a lot of population density. And while they used to have these big winter flocks when they first went out and started to learn their songs, now a lot of them are alone. There's not a lot of other honeyeaters around.
Oh.
And so when they're learning their songs in their first year, they don't have anyone to listen to. And they learn by listening. Yes.
Oh, that makes me so sad.
I know.
It's a very good-looking bird.
It's a beautiful
For those of you who are
It's
just
a
listening,
Nito.
the black, white, and yellow.
Mhm.
Yellow and black going on the feathers. Very, very pretty bird.
Yeah. And so over this 5-year study, about 12% of male honey eaters, they would wind up producing mangled versions of their songs. And they would mix it with other bird species because that's what they heard. So they would mix up honey eater song with friar birds and cuckoo shrieks and all sorts of other birds that have ridiculous names. So it ended up sounding wrong and kind of muddled. And those unconventional male singers at 12%, they were pretty consistently less successful in wooing a mate.
Yeah, they're not
Yes.
singing the right song. So, and through selection, the females are like, I only like this song and you're not singing it. So then they're not excited.
So the kind of avoidance by the females could be a few things. Obviously, they're all based in selection, but what is the motivation there? Why is this weird song a turnoff for them? So there's a couple ideas. One is that they don't recognize them as a potential partner because they don't sound like a honey eater. They're just like, who are you? Stranger. Stay away, stranger. Or they approach, but then the song takes a weird turn. It's like using a random word in the middle of a sentence, and you're like, what? You okay? So there could be that. They feel like they're unfit, or they're using the wrong cues, or it's not sexy, or whatever you want to call it.
Yeah, it would sound like a bot, like you were communicating with a bad AI.
Yes, exactly. Yes.
Like, this sounds like a trap.
Yes. Welcome to my home. Can I offer you an artichoke? What? Huh? Do you mean like a glass of water? I don't know. Anyway. So
Here's some fresh tennis shoes.
why, thank you. I'm gonna go. Yeah. So there's this weird context thing that's happening that doesn't really work. So this is an overarching, larger concern. When a species population drops that depends on social learning, do you lose species knowledge from a population when a population size declines? So this could be a much bigger problem that's an important part of looking at endangered species that we haven't even really thought about before. When population density drops, when population density drops, do you lose social knowledge? Do you lose the movement of knowledge across a species? So cultural knowledge within species is something that hasn't really been looked at too closely when it comes to declining populations. So that's a huge thing. Now, one thing that they actually did to see if there was a way they could fix this problem just experimentally is they took young birds into a captive breeding program, and they either, in some cases, they played male song recordings for them, and in other cases, they housed them next to adult males. And it appears to be working. It's very early in the testing, but it looks like these younger males are impressionable enough that they're learning via these next door singers or a tape recorded message. So this could be helpful in small bits. This could be helpful maybe by putting a speaker out in the wild for some animals that are known to winter in a specific area. But it's definitely a Band-Aid. It's not solving the bigger problem here.
But I mean, it's a Band-Aid, but perhaps it would be enough of a Band-Aid to teach the young males the songs they need to learn so that there is actually more attraction with females for mating, which could help to bolster the population sizes. Obviously, some males are the population, although they're endangered, they're still kind of going, even though they're declining. So there are some good songs out there. There's some good mates out there. And if the researchers, and maybe this is the kind of thing that they could enlist people to help to put out recorders during the breeding season, to put out speakers to play these songs so that these young male birds can learn stuff. This sounds like not like a citizen science project, if I ever heard one.
Yeah. But isn't this, I mean, Blair, do you think this has crossed the threshold with 300 individuals?
Yeah, it's a small population.
Where you stop just trying to encourage in the wild and you start thinking about breeding programs. You start thinking about captive preservation and rebuilding, like California did with the condor.
Yeah, and I think that's actually the thing I just got really excited about, is thinking about captive breeding programs. So when you're doing captive breeding programs, now this is a really good wake-up call that this needs to be part of the research when you develop a captive breeding program. Is there cultural knowledge? Is there social learning happening during the time period of when your captive breeding is happening, between when you pull eggs or hatchlings or whatever they are and when you release them into the wild? Because if there is, then you have to figure that in, or you are not going to have as successful of a program. If they didn't know this, they might pull a bunch of honeyeaters, put them in captivity, raise them up, throw them out in the wild, and then be like, why isn't the population going up?
Right. And this is the same, the whooping crane, the condors, very similar. There are dances that some birds do there are some lessons. You're right. There are some lessons that young birds need to learn from older birds to be successful as adults. Yeah. Birds need bird school.
They
my
do.
Goodness.
They're so special.
They are. What's your next bird? What's your next sad bird story?
Well, snowy plovers, they're not going to get homeschooled by their mom, because their mom, guess what? She leaving.
She's like, I'm out.
She's out. So in snowy plovers, females will often ditch out on their chicks. And so first of all, I have to explain why this happens. So there are equal numbers of female and male snowy plovers when they hatch. However, males, for whatever reason, just part of their biology, have survival advantages at all life stages. These guys live in really tough habitats. They breed on salt flats or in brackish inland lakes. They have a barren environment that puts huge pressure on them at all stages of life, but especially when they're parents. And they have temporary salt ponds where they raise their babies. Those dry up and chicks can die of thirst or starvation during that time. So it's tough to be a snowy plover. But on top of that, it's harder to be a female snowy plover. And so there is a surplus of males when they reach adulthood. There are more females than males, which flips the script.
Wait.
Or there are more males than females. Sorry.
Okay,
yes.
Good. Okay.
So that flips the script on selection. And that actually means that the females have a huge advantage in finding mates, and that they can actually play the field, because for each female, there are several males.
Mm-hmm.
So it might actually benefit them to try to have families with multiple males. There could be a benefit there because there's more males than females. So that opportunity presents itself biologically. Okay.
So in snowy plovers, it's instead of my 2 dads, it's my 3 dads, or maybe my 4 dads.
Right.
Yes.
So that exists. On top of that, let's keep in mind that when we talk about how females put all this extra energy and effort, when we've talked about that before, a lot of the time we're talking about mammals who have to carry babies with them, but these buddies can plop eggs down after a few days, and then those eggs will incubate for 20 to 30 days, and then they have to be taken care of by somebody, but it doesn't have to be the female. So actually, there is a possibility with birds for the female to lay eggs and leave. And there's also a pressure on the male to take care of babies because the females get their pick. And so if the females are selecting for males that take good care of the babies in the nest, they have the ability to make that choice, kind of pick the males who are going to make good dads. So all of this to say, the snowy plover culture is such that females are set up so that there is no evolutionary advantage to play the field. Top of that, this is a study from Max Planck Institute for Ornithology in Germany. They actually wanted to see what the decision-making process was, because the females don't always leave. Sometimes they do. Sometimes they don't. Sometimes they leave their babies to be taken care of. Sometimes they leave their babies to die, which, sorry, that's nature. That happens sometimes. So sometimes they're just cutting their losses. And so they wanted to see what the decision-making process was here. Why does a snowy plover leave her nest? This is also, I brought it because it's an uncomfortable thing to talk about, because we want to talk about moms as being the ones, right? They are the child care giver, but that's not the case in humans, and that's not the case in the animal world either. It's kind of a broad brush that we've used, but I wanted to point this out because this is also the thing that we stereotypically talk about males doing in species, but this is something that, in this case, the females are doing.
Yeah.
So what they showed through the study, they looked at 260 broods over 7 years. 70% of those broods were deserted by females. Chicks from abandoned broods did survive less often than the broods where females stayed longer, but part of the reason that kind of data exists, the reason that indicator is there, is because broods were also more likely to be deserted at the beginning of the breeding season when there weren't as many resources, and when a chick died.
Huh.
So it was as if to say,
I lost one. I'm going to lose them all. I got to go.
Yes.
I'm going to start over.
There's something wrong with this batch. I got to start over.
Yep.
Which sounds very blasé, but this is part of nature. I'm sorry. This is part of trying to figure out how to get your DNA to go, right? Is you have to make difficult decisions. And for example, boobies, they do this crazy thing where it depends on the type of, I think masked boobies actually physically kick out. They always have two chicks. They kick one out. They pick which one is more likely to survive, and they ditch the other one completely. They say, I'm putting my resources into this one. The blue-footed boobies, I believe I'm getting this right, just neglect one.
Yeah.
And it sounds really scary because we want to put our own anthropomorphic stuff on there, but ultimately, at the end of the day, they're trying to make sure their DNA makes it. Yeah. And so they have to make decisions that sound scary to us because we're putting them in our own frame. But really, that's what's happening here.
Yeah, it's interesting. We do anthropomorphize, and with birds, the brains are different, and emotions and decisions are arrived at differently than the mammalian brain, for sure. But, we like to say, oh, it's a tough decision to leave your nest or to, choose one chick over another. But it's very likely just sensory information, and the parents are making an instinctual choice.
Yep.
Based on what they see, what they smell, what they're experiencing.
I also kind of wonder if it's actually a more competitive mating environment for the females because there's less of them.
Yeah.
So it sounds like almost a little bit sideways because why would they have more competition? There's plenty to choose from. Yeah, but to get the good ones, get the to go for, if you're in charge of selection, you start to prioritize things more than if you're just lucky to find somebody. Oh, I'm just like, the numbers the other way. It's just, I'm just lucky to have found somebody. Thank you for mating with me. I'm going to mate with you and then I'm so happy that you even are here right now. Just that's enough. But if you are really being able to be choosy, then that drive to compete in the selection process means, hey, you know what? It's not working out. I need to get back in the game. And before the blossoms fall or whatever the time of the year it is, whatever, I need to have a new mate who is ready to go. And I want them to be a good one. So I need to go out there and select.
Well, and that's the other piece, right? Is who's left who's not tending to a nest, because the males are the ones that have to tend to a nest, right? So if you have to start over because you feel like this set of chicks is not going to make it, you have to start over. you have to act quickly. You also have a short mating season and breeding period. So you have a ticking clock there before you lose a whole year.
And where was the male bird? If they left, that's their job. They have one job or two jobs. But the second job is watching. If it's not there taking care of him, then I picked wrong. This is not going to be good for future generations.
Well, and this is the other thing. So it reminded me of the conversation we were having about the COVID vaccine. So there is a chance when the female leaves, there is a higher mortality rate. However, there's still a pretty high success rate. So it was making me think about, okay, yes. So there's this small group of people who are having blood clots, and perhaps they would not have a blood clot if they did not take the vaccine. But their likelihood of dying from coronavirus is much higher.
Or getting a blood clot from surviving corona is also a thing that's been happening. So it's like there's no...
So would you be better off without either? Perhaps. But with these two choices, the female's choice is they have a pretty good shot of surviving with dad. I'm going to go try to make sure that I have some chicks that survive to next year.
The one thing, I know we're not talking about the virus anymore, but the one thing about that is that the one thing about it, though, is that I know how to take precautions to avoid getting the virus. Once the shot is in the arm, the fear is that you don't have control over it. So that's why they've shut it down. If it's going to do any harm, in Denmark, I think was the first one to stop.
It's all the Scandinavian countries, several Southeast Asian countries,
they're going to
European
stop
countries. Yeah,
try to delve deeper into it to figure it out. But the element of just not knowing and controlling that the numbers don't make sense to a human.
Those plovers, though, they're just going to keep mating. They're going to keep doing what they do.
And they're controlling what they can.
And those cute little...
Exactly.
Is it the snowy plover? No, it's the killdeer that drags the arm. The mama drags her arm.
Oh,
not her arm, her wing. Drags her wings. Yes. Look at me. Come over here. I'm a decoy.
Yes. No, the snowy plovers are the ones all over the Pacific coast that run back and forth from the tide and are the reason you can't bring your dog on the beach.
Yes.
Is they're protected.
Snowy plovers are cute.
They're
adorable
birds. And it turns out their mom has
kind of brutal.
Their mom is she's a working mom. Dad's staying home with the kids. Her job is having a second family. So
she's like, I gotta go have another family right now. See you, kids. Good luck.
Yeah. What it takes to survive. Pass on those genes.
Thanks, everybody, for joining us for another episode of This Week in Science. This is This Week in Science. It really is. We're talking about science. Hey, are you enjoying the show? Please tell a friend. We love it when you do that. This is time now to come back. Justin. What did you bring to talk about?
Did I have more stories?
You don't. You do. Do you? Yeah. You tell me.
So this is an international team, 26 different authors on this single paper, including 6 of them came out of UC Santa Barbara. They just published in the journal Nature, and they kind of looked at the problems that are besetting mankind's future, and really did a pretty narrow observation on what needs to be done to protect our ocean. So the researchers identified specific areas of the ocean that could provide multiple benefits if protected. Safeguarding these regions that they suggest would protect nearly 80% of marine species. And by doing that, also increase fishing catches by more than 8 million metric tons by having preserved areas where the fish that are getting fished can be free to breed. And also prevent the release of more than 1 billion tons of carbon dioxide by protecting the seafloor bottom from trawling, which is a destructive fishing practice that I didn't realize contributed that much to carbon dioxide into the oceans, but it does. This is quote voice of Enric Sala, an explorer in residence at the National Geographic Society. Ocean life has been declining worldwide because of overfishing, habitat destruction, and climate change. Yet, only 7% of the ocean is currently under some kind of protection. And what they are encouraging is, even though there's these specific areas that would be the most useful, They know it's going to come down to every single nation, country, society having to dictate laws over the waters that they control. But what they're pushing for is getting to 30%. We're at 7% protected waters now. They want to push it up another 23% to get us to 30% protection by the year 2030 as the minimum that we need to have any shot at a sustainable future on planet Earth. So, protect the waters, everybody.
We can do this. I mean, come on. We've done so much this year for COVID research, and it takes focus, takes money, but this is something we can do.
So this is something I think about all the time is what to do about the international waters and the things that, no one has any control over. And actually something that we talk about at home all the time is, what if Aquaman was real? And it would take somebody.
Who talks about this all the time?
Me and Brian, we talk about it all the time.
Okay. I was making sure, because I'm like, I missed those conversations completely.
And specifically the other day, he was talking about Namor, who I didn't know who he was, but he's like the Marvel version of Aquaman. But anyway, the idea that there would have to be somebody with a voice living physically in the ocean to be able to come out and be like, you can't use this anymore. You got to go. You're being so disrespectful with the space that we live. You gotta go.
At the beginning,
we need Happy Feet to happen. We need that penguin with Robin Williams' voice to come out of the aquarium dancing,
we had
teaching us not to destroy the ocean. Come on.
We had it earlier this year, the end of last year. I can't remember exactly what it is. The orca that were turning fishing boats around?
Yes.
And ruining propellers and physically pushing boats out of the fishing lanes. Those voices are raising their voices as best they can for something that lives underwater.
Yeah, they need to speak English and carry a trident is the point.
Okay.
Exactly. Come on.
But they do actually have targets that are located in international waters. They have areas of the Mid-Atlantic Ridge, the Massachusetts Plateau in the Indian Ocean, Nazca Ridge off west coast of South America, and the southwest Indian Ridge between Africa and Antarctica. So there's other areas that they have pointed to in international waters that, if protected, would also massively both reduce the contribution to global warming, but also create a sustainable ocean fishery for future generations to come.
You know what would be great, actually, is instead of anything's allowed in international waters, nothing's allowed. It's like, hey,
just don't do it.
We have no control over the space, so guess what? You can't do anything but sail through it.
I like that. I like that too.
Okay, and
no one will agree to it. Okay, what's your last story?
This is a 100,000-year-old DNA was pulled out of the mud of a lake in the Arctic. This is, I think it's called Baffin Island, which is located in the northeastern side of Arctic Canada, sort of right across from Greenland territory of Nunavut and the lands of the Quiquitani Inuit. Fun fact, Baffin Island, 5th largest island on planet Earth. Huh. Did you know that?
No, now I do.
Nobody goes
Trivia.
to that island because it's freezing cold. So what they did was they did that sort of environmental DNA thing where they just have sediment and they tested DNA of what was in the sediment underneath this Arctic lake that's there. And they found evidence of a shrub. They found a shrub that doesn't grow there. In fact, the shrub is native to northern Canada, some 250 miles south. This is published in the Proceedings of the National Academy of Sciences. Sarah Kropp, researcher of the Institute of Arctic and Alpine Research at the time of the study, quoting here, "We have this really rare view into a particular warm period in the past that was arguably the most recent time that it was warmer than present in the Arctic. That makes it really useful analog for what we might expect in the future." Dwarf birch is the species of shrub that they found. It's kind of a knee-high type of a growing thing. Tends to pop up under the tundra, so you got the snowy tundra in Canada there, and then this shrub shows up everywhere, and it turns things very green, and it kind of creates a bit of a heat feedback loop, in that it kind of the ice around it, the snow around it, will melt, and of course use water, and that's how it survives. But it doesn't grow anywhere past a certain point in the southern part of the Baffin Island in the Canadian Arctic, because it's just too cold. Cody voice again. It's pretty significant difference from the distribution of tundra plants today. Again, that's Crump. It's actually currently a postdoctoral in the Paleogenomics Lab, University of California, Santa Cruz. So one of the things that, we're talking about here, though, is this is from 116 to 125,000 year old sediment where this shrub was in this location. And when we get into cooler periods, it takes like thousands of years as these arcs of temperature rise and fall. We're doing things very quickly now. So one of the points that this study sort of brings up is, yes, there are plants that can survive other places, but how do they move there? And this is something that can survive if the temperature that we're going to see at the end of the century is 9 degrees Fahrenheit, 5 degrees Celsius higher than it was the pre-industrial, because the Arctic is warming faster than the rest of the planet. These are very, very achievable. Does where the plant is now make it inhabitable for this shrub? Will it transport? Will it begin to move? If it can't move in time, it dies. And so this is just one shrub, right? That has moved, has backed off 250 miles of territory, but doesn't have another 250 miles of territory to its south that it can live in either. It's in this band where its biome is secure, and it can function and it can exist. So this is one specific shrub, but the implications of this are for every form of tree and plant life on this planet. That if you can see a 250-mile change in territory of its ideal habitable zones, that could be absolutely devastating.
That's pretty interesting, though, that these plants, right? Maybe someday soon, the plants of today will be competing against their ancestors. No.
No. I mean, but the point is, like, can you picture like having to take okay, all right, we have to move the desert cactus 250 miles north
50
and
miles
replant
north.
it,
Yeah,
or 50 miles or whatever. Like start having to move all of the plants. That's a lot of work, people. Come on. I know humans. We're much lazier than moving all of the plants.
The plants move themselves.
We
lazy. Force the situation that's causing the moving of the plants.
But it's going to happen too quick for the plants to do it. So what I'm saying is, if we're too lazy to move all of the plants 100 miles north or whatever it takes, we should just stop warming the planet.
Yeah, that would be
Good
easier.
Point. It would be easier. Let's just not do it, everybody.
Let's do
okay?
the easy thing and save the hard thing for later.
Yes.
You think it was Kennedy who said that? We're going to do the easy thing. Put off doing hard things till some other time. We should do it that way.
Sometimes the easy thing is the right thing.
Why do Irish eyes cry?
Why do any eyes cry? Because they're sad.
Well, it's because of your lacrimal glands.
Yeah.
I mean, come on. Lacrimal glands. Yes, and researchers have decided that, yes, we need to grow lacrimal glands from stem cells in a petri dish. Of course we do. To understand exactly how they work and what genes are involved. This is important to understand this. Actually, it is very important. The researchers say that the lacrimal glands and the act of crying, the tear production, is not really fully understood, like so many other things. But there are disorders like dry eye and dry mouth that are genetically related, that if we could figure out how to treat them, if we could understand how this part of the eyes work, it could help out a lot of people. Anyway, researchers took the lacrimal glands from mice, stem celled them up, grew them in dishes, and then looked to see how they grew and whether they could grow them in dishes. That was the main point of their work was, can we grow them outside of an animal? And yes, indeed, they could. They were able to get full organ growth. They found all the cell types. They did have to mix up their chemical cocktail a little bit to get the glands to work. But when I say get the glands to work, they made these tear glands cry in a dish with the addition of norepinephrine. So in case you didn't know, chemical signal for tear production in your eyes is norepinephrine. Now you know. So the researchers were able to actually get tear production to occur in a dish. Then they used knockout technology to knock different genes out to see how that affected tear production. They found one gene called PAX6 completely dismantled tear production. Tears were not formed. The lacrimal glands didn't do what they needed to do. It was a big shame, a shamble, no tears. The glands had no tears. I have no tears for you, they said to the researchers. Well, they didn't really, but the end of the story is now they have some direction to go in the study of the genes that are related to tear production, the development of the lacrimal glands. And there is a syndrome, which is called Sjogren's syndrome, which does result in dry eyes, dry mouth, and a lot of pain for people because of it. And it is related to a mutation in the PAX6 gene, apparently. So when Irish eyes are crying, norepinephrine has been released, and the lacrimal glands are producing tears.
Right when at the beginning of the story, in the end of the intro of the story, when you're like, "And did they do it?" I would just kind of wish you had said no. Like, as it gave me this, like, we should do like a null report version.
Did they do
of
it?
the show.
No. Nope, didn't work.
So researchers studying if they could combine these two elements and create this other thing, and it didn't work.
Oh my gosh. Okay,
Just like a whole
researchers.
episode.
Okay, researchers who are studying things, please send us your null results.
Yes, please.
Can you send us your null results? If you're a researcher and you have null results, we'll make a collection of
They don't
null
even need
results.
to be current. We'll make an exception.
Yeah,
They don't
we
have
will-
to be
One
current.
of my
make it-
best friends
to
got
get
her
this started.
PhD on a null result.
Send
Yes.
it in.
Yes,
But
it was
yes.
it was fascinating when I saw her defense, and it was very interesting, actually.
Yeah. Oh, those
But
are-
I
you have to have
think
them.
that will
Yeah.
be a fun episode. They were looking at this and they were asking this question and they did all these things, and did they find it? No.
No, apparently there's no mechanism there whatsoever. It's just a correlation.
Oh, I can't wait. This will be
On
a
to the
great
next.
episode. I love it.
Well, because there are, I mean, in honesty, there are way more null results than there are
Positive, yeah.
positive hits or really enlightening ones.
Yeah.
And all of those do lead to a narrowing. It's part of the whole scientific process. It's all part of it.
Anyhow, my final story. Oh, you people with green eyes. I'm just thinking green eyes for Irish, but it's not necessarily. Brown eyes is probably very common for the Irish. Researchers at King's and Erasmus University Medical Center in Rotterdam have identified 50 new genes for eye color. So we all, not all of us, but I mean,
50?
when in school,
50.
in school, I learned genetics using the Punnett square and dominant and recessive genes for, say, brown eyes. You know, big B's brown eyes, little B's blue. 1 out of 4 is blue. Oh, look how that works. Very simplistic, but that gave me my understanding of modern genetics and eye color. It's a bit more complex than that, just a little bit. It was published in Science Advances and looked at over 195,000 people across Europe and Asia. hopefully this isn't just a study looking at eye color, but will help understanding of eye disease and pigmentary glaucoma, ocular albinism as well, where eye pigment is actually related to disease in the eye in some way or another. Additionally, they found that eye color in Asians with different shades of brown is genetically similar to eye color in Europeans that range from dark brown to light blue. So same genes,
So there's
different
other
shading,
genes involved.
different pigment. Yeah, really fascinating there. So previously it had been discovered
green
that
and yellow and like
Yellow.
yellow. I have a friend who's got just yellow eyes. There's no other. Yeah.
Right, and that's definitely unique. So how many genes are involved in that?
And then sometimes have heterochromia, so they have two different colored eyes, right?
Right. Yeah. So anyway, eye color, it's not just two genes, as some scientists long time ago previously thought. Then it was a dozen genes. Now it's over 50. Over 50 genes for eye colors. So your beautiful, beautiful ocular sockets are pigmented very complexly. And that complexity is beautiful.
If I may, I think there's still certain things that are heavily dominant, right, within all of that.
They're probably, it depends on how they're linked. Right?
My dad's entire side has these extremely dark brown eyes, like so dark they almost look black, and I have them. And I think that there's something there because, all of these Basduriches all the way down, a lot of them married people with light eyes. My mom has light eyes. Lots of people have light eyes in that family, but their kids pretty much always ended up with these really, 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 really dark eyes. And so, it's very anecdotal, but I think that, the Punnett square might be an oversimplification, but there's definitely some interesting stuff going on there with dominance with the eye colors too. Just like with hair too, like curly hair is dominant, but you can also have weird mixtures of hair. And
two out of my 3 kids have blue eyes.
And you have brown eyes. No, green eyes?
I think I have the
hazel.
I have the both.
Both.
So my pops had blue eyes, and my mother has brown eyes. They're kind of hazel. They've got a little green.
Hazily. Yeah.
Too.
So
I
was going to say from the abstract for this paper, which is in Science Advances, they say we identified 124 independent associations arising from 61 discrete genomic regions, including 50 previously unidentified. We find evidence for genes involved in melanin pigmentation, also associations involved in iris morphology and structure. And then they go on to discuss the Asian versus European eye coloration. Collectively, they say that their results explain 53.2% of eye color variation using common single nucleotide polymorphisms. So there is still more to be discovered. If their findings only explain about 50%, it's only halfway there. There's so much more to find. More associations needed. More study of eyes. Let researchers gaze deeply into your eyes. Have we come to the end of another episode? I hope all of your eyes are on your calendars and marking them for April 17th, Saturday, April 17th, for the TWIS DTNS crossover whip episode. Science times technology.
Anyway, if you have something that you would like us to talk about related to the cross of science and technology, it could be a specific story, it could be a larger topic, please let us know. You can, we're going to tell you how to find us in a second in all the myriad of ways, but any of those ways, tell us if there's something that you want us to bring to the show.
Yes, thank you. But mark your calendars. More details will come. TWIS DTNS crossover. For some reason, I really want to play it out.
Are we gonna be fighting?
No,
Yeah.
Is there a monster truck involved?
It's like a monster truck rally, but science and technology. So it's going to be amazing.
Wait, what day of the week is that? Is that a Saturday?
It's a Saturday.
Saturday!
Yes. Science, technology. Thank you, everyone, for listening to the show. We are so glad that you were here with us tonight. Thank you for joining us tonight. It's time for me to give shout outs. Thank you to my co-hosts. Thank you, Justin. Thank you, Blair. Thank you, Fada, for all of your help on social media and for show notes. Thank you, Gord, for your help in the chat room. Identity4 for recording the show. Rachel for all of your assistance. And I would like to thank our Patreon sponsors. Thank you to Karen Tazi, Woody MS, Andre Bassett, Chris Wozniak, Dave Bunn, Vegard Chefstad, Hal Snyder, Donathan Stiles, a.k.a. Don Stylo, John Chio, Lee Guillaume, John Lee, Ali Coffin, Gaurav Sharma, Shubru, Darwin Hannon, Donald Mundis, Stephen Alberon, Daryl Myshak, Stu Pollock, Andrew Swanson, Fredus104, Skyluke, Paul Ronovich, Kevin Reardon, Noodles Jack, Ryan Carrington, Matt Bass, Joshua Fury, Sean and Nina Lamb, John McKee, Greg Riley, Mark Hassenflow, Gene Tellier, Steve Leisman, a.k.a. Z. McKen Hayes, Howard Tan, Christopher Rappin, Dana Pearson, Richard Brendan Minish, Johnny Gridley, Kevin Railsback, Flying Out, Christopher Dreyer, Mark Massaros, Artyom, Greg Briggs, John Atwood, Robert Coburn, Rudy Garcia, Dave Wilkinson, Rodney Lewis, Paul, Rick Ramus, Matt Sutter, Phil Shane, Kurt Larson, Craig Landon, Mountain Sloth, Jim Drapeau, Sarah Chavis, Sue Doster, Jason Olds, Dave Neighbor, Eric Knapp, E.O., Kevin Parachan, Aaron Luthen, Steve DeBell, Bob Calder, Marjorie, Paul Stanton, Paul Disney, Patrick Percararo, Tony Steele, Ulysses Adkins, Brian Condren, and Jason Roberts. Thank you for all of your support on Patreon. And if you would like to support us on Patreon, head on over to TWIS.org and click on that Patreon link. On next week's show,
we will be back Wednesday, 8 p.m. Pacific time, broadcasting live from our YouTube and Facebook channels and from TWIS.org/live.
Hey, do you want to multitask? Do you want to listen to us as a podcast? Get some science fix while you do the dishes, bake a cake, go for a walk. Just search for This Week in Science wherever podcasts are found. If you enjoyed the show, get your friends to subscribe as well.
For more information on anything you heard today, and show notes, links to stories, that sort of thing, they're all available on our website. Just go on over to www.twis.org, and you can sign up for a newsletter if you like.
Oh, that's right. You can also contact us directly. Email Kirsten at kirsten@thisweekinscience.com, Justin at twistminion@gmail.com, or me, Blair, at blairbaz@twis.org. Just be sure to put TWIS, T-W-I-S, in that subject line, or your email will be spam filtered into a tear duct, I guess.
The smallest tear duct you've ever seen. You can also ping us on Twitter, where we are at TWIS Science, at Dr. Kiki, at JacksonFly, and at Blair's Menagerie. We love your feedback. If there's a topic you would like us to cover or address, a suggestion for an interview, a haiku that comes to in the night, please let us know.
We'll be back here next week, and we hope you'll join us again for more great science news.
And if you've learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's this This week in science. This week in science. This week in science. 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 This week in science. This week in science. This week in science. 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 I've got one disclaimer, and it shouldn't be news, that what I say may not represent your views, but I've done the calculations and I've got a plan. If you listen to the science, you may just yet understand that we're not trying to threaten your philosophy. We're just trying to save the world from jeopardy. 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 And this week in science is coming your way. So everybody listen to everything we say, and if you use our method instead of rolling a die, we may rid the world of toxoplasma gondii. Cause it's this week in science. This week in science. This week in science. 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 This week in science. This week in science. This week in science. 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 Got a laundry list of items I want to address, from stopping global hunger to dredging Loch Ness. I'm trying to promote more rational thought, and I'll try to answer any question you've got. But how can I ever see the changes I seek when I can only set up shop 1 hour a week? This week in science is coming your way. You better just listen to what we say, and if you've learned anything from the words that we've said, then please just remember it's all in your head. Cause it's this week in science. This week in science. This week in science. 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 This week in science. This week in science. This week in science. 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 This week in 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 science.